Hello everyone. I welcome you at YouTube channel of Advanced Multiple Incorporation. Advanced Multiple actually a knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha. I'm working as a scientific executive with the Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Both of these organizations are located in Mississauga, Ontario, Canada. Okay, well, I'm going to share my knowledge, my experience, and my information under the title of Chemical Characterization of Preservatives used in Pharmaceutical Industry. For this purpose, I collected the information from different data sources like PubMed, Medline, Scoopers, Google Scholar, National Library of Medicine, MBAS, Directory of Open Access Journal, ProQuest, CNKI, that is stand for the China Network for Knowledge Infrastructure, some of information collected from Crossref and ResearchGate. Okay, before moving forward, I would like to talk a little bit about the chemical characterization of preservatives. The substances used to preserve medicine from spoilage by preventing the growth of microorganisms called preservatives. It is very simple definition, description of preservatives. Then preservatives should be highly water soluble, and they should be free from odor, color, toxicity, irritation, and sensitization. They should be compatible with other ingredients and packing material and container a wide spectrum of anti-activity against bacteria, yeast, mold are required. It should also have to protect the product from opportunistic pathogens. Now, I would like to talk a little bit about chemical characterization and classification. Preservatives are mostly used in oral preparation, topical formulation of ophthalmic dosage forms, and parenteral products. So, wherever we use the water as major component, the pharmaceutical dosage forms they need the preservative to protect it from bacterial spoilage. So, preservatives are chemically classified into seven groups. Why we classified it? Because we have to understand it. We have to make it more understandable and conceivable. We classify on behalf of different characteristics. Some product they are, or some entities, or some services, or some product they are classified on behalf of their physical nature, chemical characterization, or on behalf of their color, or on behalf of their source, maybe natural, maybe synthetic, maybe semi-synthetic. But here, in case of preservative, we have classified the preservative on behalf of their chemistry, their chemical nature, their formulation. So the first class is acid and esters, then alcohol, then aldehyde, bicarbonate, heavy metals, phenols, and paraben. These seven classes of preservative we are going to discuss one by one. The first class here we are going to discuss is the acid and esters. Benzoic acid. The natural source of benzoic acid is gum benzene, which come from certain tree bark. Benzoic acid can also be made by synthetic means. It is colorless crystalline solid with chemical formula of C6H5COOH. Its salts are used as a food preservative. It is advisable to use limitedly with maximum pH of 5. Benzoic acid and salicylic acid used in combination for topical medication used for the treatment of the skin irritation, inflammation caused by burn, insect bites, fungal infection, or eczema. 0.05 to 1% concentration of benzoic acid suitable for oral preparation, 0.25% for the topical, and it is also used for the injectable or parenteral preparations. Optimum activity is obtained at the range of pH 2.5 to 4.5. Benzoic acid inhibits the growth of the mold, yeast, and some bacteria. The mechanism starts with the absorption of benzoic acid into the cell membrane. Once it's absorbed, then it starts its activity and eventually kill the microorganisms. Benzoic acid is a constituent of white peel ointment, which is used for the treatment of the fungal skin disease such as tinea or the tinea, ringworm, and athlete foot. Sorbic acid is white crystalline powder, relatively tasteless and odorless. Sodium sorbate is solid form, is unstable and very rapidly undergoes oxidation on exposure to atmospheric oxygen. It is therefore not produced on the industrial scale. This is widely used as acid or its potassium salt for the topical use. Potassium sorbate and sorbic acid possess antifungal and limited antibacterial properties. It is effective at the pH of 4 or below. It is mostly used in gum syrup. Sorbates are generally used at the concentration of 0.025 to 0.10%. Okay, alcohol. Benzyl alcohol is bacteriostatic, used at concentration of 2%, used up to 5% oral, up to 3% topical, and most effective at the pH of 5. It is used as preservative in injectable and topical medication. Chlorbutanol is bacteriostatic, used at concentration of 5% in injections and eye drops, 0.3% topical, and 0.6% ophthalmic or eye preparations. It becomes ineffective if pH is above 5.5. Isopropyl alcohol is a preservative and natural disinfectant commonly used in mouthwash and lotions. Ethanol kills organisms by dissolving their lipids and by denaturing their proteins. It's effective against bacteria, viruses, and fungi. Phenyl ethanol is used in concentration of 0.25 to 0.5%, having greater activity against gram-negative bacteria. Okay, well, aldehydes. Glutaraldehyde is allowed as a preservative in cosmetics. It is used as an acidic 2% and greater aqueous solution. It is required for core, liquid, and chemical sterilization. Formaldehyde are used as reactive antimicrobial for controlling microorganisms with protein and peptide. Poor penetration and tendency to polymerization reduce the actual usage as preservative. It is highly toxic, therefore not commonly used as preservative. Next class of preservative is bigonite. Bigonite have two subcategories, chlorhexidine and alexidine. Polyhexamethylene bigonites. So chlorhexidine exhibit the greater extent of antibacterial activity at the pH of 7 to 8 and used topically and parenterally. It is used in the digluconate form and act by destroying the semi-permeable layer of the cytoplasmic membrane and produce its antimicrobial activity mainly against cocci and gram-positive bacteria. Gram-negative bacteria as well as fungistatic activity. It is well tolerated and non-toxic, used in important pre-operative antiseptic preparation. Laxidine is used to be gentle and potent antibacterial agent used in the contact lens. Polyhexamethylene biguanide, it has a broad spectrum and activity against gram-positive and gram-negative bacteria. It has low toxicity and is implied as an antimicrobial agent in various ophthalmic products as well as contact lens solution. So next category of preservative is the heavy metals. In heavy metal, we use the mercurals. Mercurals are bacteriostatic and applied as preservative, topically antiseptic, and used in parenteral preparation. Use of these compounds has declined considerably due to the concern about the mercury toxicity and risk of hypersensitivity of local irritation. Next class of preservative is the phenol. Phenol are widely used as a disinfectant and preservative. They have bactericidal activity but generally are not sporicidal. Phenol act as preservative in some multi-dose parenteral products. 0.2% of phenol is suitable bacteriostatic in insulin injection at the pH of 5. Next class of preservative is paraben. The paraben are used as preservative and solvent of many drug medicines, especially in cream, drop, suspension, and syrup. These are most commonly used because of their low toxicity, effectiveness against mold and yeast. The paraben are esters of para-hydroxybenzoic acid. So butyl paraben commonly used in combination with methyl paraben. It is also used as antiseptic. Methyl paraben is completely absorbed through the skin after ingression, or it is hydrolyzed into the para-hydroxybenzoic acid, and metabolites are rapidly excreted in the urine. There is no evidence of accumulation and also used in inhalation, nasal, and rectal preparation. Propylparaben often used in combination with the methyl paraben. So at last, in conclusion, we can say that the chemical characterization of preservative is primarily important to assure the compatibility, suitability, and acceptability of product. In polypharmacy practice, The least quantity of most relevant preservative is preferred in the treatment of patients with multiple clinical syndrome that may potentially help to improve the efficacy of drugs and improve its stability too. At last, I appreciate the kind help and support, and I salute and greet to my colleague, my team, my coworker, Sherry Bajkot, Wolfgang, Daniel, Dustin Hawker, Marwa Al Maghribi, Teddy Merrill, Noreen, Kunza, Robert, Ashtiaq, Arslan, and Amin. They helped me a lot to make this video today. I also acknowledge the kind help and support of Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery and Technology Development Incorporation. As I told in the start of the video, like both of these organizations are located in Mississauga, Ontario, Canada. At last, before finishing and closing the video, I appreciate your kind help and staying with me, watching the video. Thank you very much. See you soon. Take care. Bye now.